The discovery of microRNAs mRNAs by Gary Ruvkun and Victor Ambros fundamentally changed the understanding of gene regulation. Prior to their work, it was believed that most genes were regulated primarily by proteins. However, in the early 1990s, they uncovered a new class of molecules, microRNAs, that regulate genes in a completely different way. MicroRNAs are tiny RNA molecules around 22 nucleotides long that do not code for proteins like most RNA. Instead, they bind to messenger RNAs, mRNAs, and are responsible for carrying genetic information from DNA to ribosomes where proteins are made. By binding to specific mRNAs, mRNAs prevent the production of certain proteins, essentially turning off the corresponding genes. Ruvkun and Ambros first made this discovery while studying the development of the Caenorhabditis elegans worm. They found that two genes, Lin4 and Lin14, were controlled by mRNAs regulating key stages in the worm's development. Ruvkun later showed that this regulatory mechanism was conserved across many species, including humans, and that mRNAs are critical in controlling cell growth, differentiation, and death. This discovery opened up a new field of research, revealing that mRNAs play important roles in various biological processes and diseases such as cancer, heart disease, and neurodegenerative disorders. The groundbreaking nature of this research was recognized with the 2024 Nobel Prize in Physiology or Medicine. Gary Ruvkun's initial research into microRNAs mRNAs began with a focus on understanding how genes control the timing of development in the roundworm Caenorhabditis elegans. In collaboration with Victor Ambros, they studied two specific genes, Lin4 and Lin14, which were known to play crucial roles in the worm's development, but whose regulation was not fully understood. In 1993, Ambros discovered that Lin4 did not code for a protein, unlike most genes. Instead, it produced a tiny RNA molecule, which turned out to be the first identified microRNA. Ruvkun then showed that this mRNA from Lin4 regulated the expression of the Lin14 gene by binding to its messenger RNA, mRNA, effectively stopping Lin14 from being translated into a protein. This binding prevented the normal functioning of Lin14, affecting the developmental timing of the worm. This discovery was remarkable because it introduced a completely new concept of gene regulation, one that did not rely on proteins but on small, non-coding RNA molecules like mRNAs. Until then, the understanding of gene regulation was largely centered on proteins acting as transcription factors, but mRNAs revealed an entirely new layer of control over gene expression. Later, Ruvkun's lab discovered a second mRNA, Let7, which showed that this regulatory mechanism was conserved across different species, from worms to humans, indicating its evolutionary importance and relevance to many biological processes. The discovery of microRNA mRNA regulation by Gary Ruvkun and Victor Ambros revealed not only a novel mechanism in Caenorhabditis elegans, but also an insight that this mechanism is conserved across a wide range of species, including humans. This conservation means that mRNAs regulate gene expression in the same way across different organisms, indicating that this regulatory process is fundamental and has been preserved through millions of years of evolution. After identifying the first mRNA, Lin4 in C, elegans, Ruvkun and his team discovered a second one, Let7, in 2000. This mRNA also blocked the translation of a target gene, and what made Let7 particularly significant was that it was found to function in a similar way across a broad spectrum of species, from worms to flies, fish, and even humans. This demonstrated that mRNAs are not just a peculiarity of worms, but are an essential part of the genetic toolkit used by many multicellular organisms to regulate gene expression. This discovery of mRNA conservation across species indicated that mRNAs play a critical role in fundamental biological processes, including development, growth, and cellular differentiation. Since mRNAs are involved in gene regulation in diverse organisms, they are integral to processes that affect normal function and disease in humans, such as cancer and heart disease. The The fact that this mechanism has been conserved across such different species suggests it is highly efficient and necessary for survival and development. The discovery of Micrornis myrnas by Gary Ruvkun and Victor Ambros has profound medical implications, particularly in the understanding and treatment of various diseases. These small RNA molecules regulate gene expression by binding to messenger RNA mRNA and preventing the production of specific proteins. This mechanism is critical for normal biological processes, but when disrupted, it can lead to diseases such as cancer, heart disease, and neurodegenerative disorders. Here are key medical implications of myrnas. Cancer myrnas play a significant role in controlling cell growth and division. Abnormal expression of specific myrnas has been linked to cancer, where they can act as tumor suppressors or oncogenes. For example, in certain cancers, the loss of myrnas that suppress tumor growth leads to uncontrolled cell proliferation. Heart disease myrnas are also involved in the regulation of cardiovascular functions, including heart muscle development and response to stress. Dysregulation of specific myrnas is associated with heart failure and other cardiovascular diseases. Researchers are exploring myrna-based therapies that target these dysregulated molecules to restore normal heart function. Neurodegenerative disorders, myrnas are crucial for neural development and function. Alterations in their expression have been linked to neurodegenerative diseases like Alzheimer's and Parkinson's. Understanding these myrna pathways may offer new therapeutic strategies to slow or prevent the progression of such diseases. Nickel applications, myrnas are being used as biomarkers for disease diagnosis and prognosis. For instance, in oncology, myrna profiles can help in identifying specific cancer types and predicting patient outcomes. Additionally, myrna-based therapies which involve either blocking harmful myrnas or restoring beneficial ones are currently in clinical trials for conditions such as cancer and heart disease. Overall, myrna research is opening new avenues for diagnostic tools and therapies, potentially transforming the way many diseases are treated. The discovery of Micrornis myrnas has led to significant advancements in clinical applications, particularly in diagnostics and therapeutic development. Here are the key clinical applications of myrnas. Diagnostics and disease biomarkers. Myrnas are emerging as reliable biomarkers for diagnosing and monitoring various diseases, especially cancer. Different types of cancer, such as breast, lung, and prostate cancers, show unique myrna expression profiles. By analyzing these myrna signatures in patient blood samples or tumor tissues, clinicians can identify cancer types more precisely and detect the disease at earlier stages. For example, myrna panels are being used to classify tumor types and predict how aggressive a tumor might be, leading to more personalized treatment plans. Prognostic tools. Myrnas also provide valuable prognostic information. In cancer treatment, myrna expression levels can predict patient outcomes, such as their likelihood of relapse or response to specific treatments. This allows for more targeted therapies based on an individual's myrna profile. RNA-based therapies targeting myrnas are currently in development in clinical trials. These therapies work by either inhibiting harmful myrnas or replacing missing beneficial ones. For example, in some cancers, myrnas that normally suppress tumors are underexpressed. By delivering synthetic versions of these myrnas, researchers aim to restore normal gene regulation and slow tumor growth. Conversely, harmful myrnas that promote disease can be silenced using myrna inhibitors, such as antagomers, which block their activity. Cardiovascular disease treatment. In heart disease, myrnas are involved in controlling heart muscle development and stress responses. Myrna therapies are being explored to treat heart failure by restoring normal heart function. For instance, myrna 21 has been shown to be involved in heart tissue fibrosis, and targeting this myrna could potentially reduce tissue damage after a heart attack. Other diseases beyond cancer and heart disease, myrnas have potential therapeutic roles in treating autoimmune disorders, neurodegenerative diseases such as Alzheimer's and Parkinson's, and viral infections. By targeting specific myrnas, researchers hope to modulate immune responses, slow neurodegeneration, or inhibit viral replication in diseases like hepatitis. The development of mRNA-based diagnostics and therapies is still ongoing, but early results from clinical trials are promising. These applications represent a new frontier in precision medicine, offering more personalized and effective treatments for a wide range of diseases. In 2024, Gary Ruvkun, along with his collaborator Victor Ambros, was awarded the Nobel Prize in Physiology or Medicine for their groundbreaking discovery of microRNAs mRNAs. This prestigious recognition highlights the transformative impact of their work on understanding gene regulation. The Nobel Committee acknowledged that their discovery revealed a new principle in biology, showing that small RNA molecules play a crucial role in turning genes on and off by binding to messenger RNAs and mRNAs. This mRNA-based gene regulation mechanism is essential for normal development and has implications for diseases such as cancer, heart disease, and neurodegenerative disorders. The Nobel Prize was also awarded because of the broad significance of mRNAs across species. The team's discovery in Canorhabditis elegans revealed that this mechanism was conserved across many organisms, including humans, making it a fundamental biological process. Their work has opened new avenues in both basic biological research and clinical applications, influencing cancer diagnosis, therapy development, and even heart disease treatment. This Nobel Prize honors a discovery that has had widespread scientific and medical relevance as mRNAs continue to be at the forefront of research into disease mechanisms and new treatments.